Collect the soil sample with a small spoon by digging to about 1 centimeter in depth. Cut some leaves off a plant and place them in a plastic bag.
Pour 10 milliliters of sterile saline solution into a screw cap tube. Add 1 gram of soil to the solution. Follow a similar procedure for the leaves of the plant. Shake vigorously and let the tube sit for 10 minutes.
Take some glass beads and pour them into a beaker. Pour ethanol on top of the glass beads so that they are completely soaked. Shake well and drain the beads.
Rinse the beads twice with distilled water. Drain the excess water.
Take a Petri dish that contains the growth medium. Label the bottom of the dish soil 10 to the power of -2. With a Pasteur pipette, transfer 5 drops of the solution containing the soil sample directly to the Petri dish. Avoid carrying excess solid material. Add around 10 glass beads to the dish and shake thoroughly to spread the liquid all over the surface until you notice that the surface is dry.
Take a second Petri dish that contains the growth medium. Label the bottom of the dish plant 10 to the power of -2. With a pipette, transfer 5 drops of the solution containing the plant sample directly to the dish and repeat the process described previously.
Take 2 test tubes and label them soil 10 to the power of -4 and plant 10 to the power of -4. Pour 10 milliliters of saline solution into the test tubes. With the new pipette, transfer 5 drops of the solution containing the plant sample into one tube and the same amount of soil sample into the other. Shake both tubes well.
Take the third and the fourth Petri dish that both contain the growth medium. Label them soil 10 to the power of -4 and plant 10 to the power of -4 respectively. With Pasteur pipettes, transfer 5 drops of both solutions containing the soil and the plant sample to the Petri dish. Add around 10 glass beads to the dishes and shake them thoroughly to spread the liquid all over the surface until you notice that the surface is dry.
Leave the 4 Petri dishes in a warm place for 48 hours.
Take out the dishes, count the number of colonies in each sample.
On the plant 10 to the power of -2 plate, we could detect 1 colony, and on the plant 10 to the power of -4 plate, there were 4 colonies and 2 species. 4 colonies means 40,000 bacteria.
The plate soil 10 to the power of -2 had too many colonies that could not be counted. On the plate soil 10 to the power of -4, we first marked the colonies with a marker and counted 3 species and about 700 colonies, which means about 7 million bacteria.